Hello everyone, I am honored to share the paper that our team published in the iMeta journal, which entitled Duck Gut Metagenome reveals the microbiome signatures linked to intestinal regional, temporal development, and rearing condition. Drs. Ma Lingyan and Liu Wentao of the Zhejiang Academy of Agricultural Sciences are the first authors, and Professor Lu Lizhi, Yang Hua, and Xiaoying Ping are the corresponding authors. As we all know, China is the world's largest producer and consumer of waterfowl farming. The total value of waterfowl production has exceeded 100 billion, among these, duck farming accounting for 74.3% of world production. The duck gastrointestinal tract harbors an abundance of microorganisms that play an important role in duck health and production, such as nutrients digestion, immune system development, and feed efficiency improvement. In the present study, a total of 375 gastrointestinal samples were collected, which including Heilongjiang, Zhejiang, and other 11 provinces. These samples could be divided into 4 breeds, 5 intestinal segments, 5 different growth ages, including 3 to 70 days, and 2 rearing system, including with water and without water system. Based on these samples, shotgun metagenomic sequencing were performed for duck microbial gene catalog construction and metagenome assembling. Firstly, we obtained a non-redundant GIT microbial gene catalog with 24,602,722 genes and reconstructed a total of 4,437 bacterial and archaeal genomes and metagenome-assembled genomes. A different trend in gut microbiome diversity along the digestive tract, as alpha diversity was highest in the small intestine but nearly identical in the cecum and colon. Specific microbial taxa show significant enrichment patterns in specific intestinal segment regions. Next, the comparison of functional potential showed that the small intestine microbiome appeared to be primarily involved in cofactor, vitamin, and amino acid metabolism. On the other hand, the large intestine microbiome exhibited a stronger association with lipid metabolism and functions related to protein folding, sorting, and degradation. Furthermore, we compared the regional signatures of C enzymes in the duck gut microbiome, and found that similar C enzymes are encoded by different bacterial species in different intestinal segments. The developmental trajectory and maturation of the duck microbiome further showed that the microbial diversities were increased during duck development, peaking at day 14 and then remaining stable or decreasing slightly thereafter. Dominant phyla were firmicutes, proteobacteria, bacteroidetes, fusobacteria, and actinobacteria. Interestingly, we found that on day 3, the identified major genera included Klebsiella and Escherichia, reflecting the short-term exposure to the environment and the initiation of gut microbial communities. On the other hand, the key butyrate-producing bacteria, such as Blachia and Butyroscoccus, were consistently observed during middle growing stages, suggesting a transition of gut environment towards a healthier stage. For the gut microbiota function, in day 3, the prominence of sulfur metabolism, along with glutathione and glutamine metabolism. For day 7, nutrient uptake and energy metabolism were more abundant. In day 14, carbohydrate metabolism was enriched, while in day 42 and 70, amino acid and fatty acid metabolism become more enriched. Without water condition exhibited significant enrichment in pathways related to carbohydrate, amino acid, lipid, energy, and xenobiotic metabolism, with water condition influenced pathways associated with cofactor and vitamin, glycan, terpenoids, and polypeptides, as well as nucleotide metabolism. Furthermore, in the WW duck microbiomes, we observed a reduction in the sucrose phosphotransferase gene, while the sucrase gene and glucose 6-phosphate isomerase showed increased abundance, and with increased abundance of LPXA, LPXB, LPXC, and LPXD. For antimicrobial resistance, we found that the gut microbiome with water ducks had significant beta diversity and a slight increased Shannon diversity of ARGs. And major phyla actinobacteria, Bacteroidota, firmicutes, and proteobacteria were mainly responsible for the card encoding. In conclusion, this study indicated the duck gut metagenome reveals the microbiome signatures linked to intestinal regional, temporal development, and rearing condition, further providing theoretical basis for microbial research and poultry production. Thanks for watching, and thanks for the iMeta journal.